I'm here with Jonathan Vasquez from the Biosciences building in UTEP. Jonathan, what can you tell me about how DNA works in the human body?
Okay, well, like the way that I personally like to think about DNA is that it's very much like a blueprint. So when you take a look at the building, there's a blueprint behind that that's gonna dictate what the architecture of that building is gonna look like, how many compartments, how many rooms it's gonna have, what kind of structural framework that's made out of a particular type of concrete or steel or metal. And that's kind of like the way that DNA is. DNA is the blueprint of your body, of every single organism that's on the planet has a specific blueprint, all the building blocks that are required for it. And DNA works as that blueprint that's going to dictate everything that makes you who you are.
According to Peter J. Russell, the author of Epigenetics, A Molecular Approach, the central dogma of biology states that DNA is transcribed into RNA, which is then translated into proteins, which the body uses and expresses in its phenotype. And what can you tell me about epigenetics and how it applies to DNA?
Okay, well, DNA regulation is basically regulated by all these things like enhancers, promoters, silencers that are located on the gene sequence. Epigenetics is just another level of gene regulation that it involves methylation tagging. So enzymes will start adding methyl groups to DNA segments, either silence or repress them. And this tagging works and is influenced primarily on how you live your life. So all the influences that are involved in, for example, whether you decide to drink alcohol or take drugs or live a very healthy lifestyle, are going to affect the methylation patterns that are found in your DNA. And basically, epigenetics runs really deeply because, for example, a lot of the choices that your parents made on how they left out their life, a lot of those taggings get passed on to you. And then whatever life choices you make get passed on to the next generation because of these epigenetic tags.
In a pair of identical twins that share the exact same segment of DNA, methylated regions determine whether certain phenotypes are going to be expressed in the individuals. Now, this is determined based on whether they live lifestyle choices such as smoking or eating healthy, or it can be based on environmental factors that come into play. I'm also here with Gabby Franco, who also works in the Biosciences building. Gabby, can you tell me something about how histone modification affects epigenetics in humans as well?
Histone modification is another mechanism that can result by environmental factors and also affects if a gene is expressed or not. A histone is basically a spool that the DNA wraps around. If DNA is too tightly wrapped around the histone, the ribosome will not be able to reach this segment of DNA. That gene will not be expressed.
As a direct result of patients who formed diseases and research based on their DNA analysis, the scientific community can conclude that diseases can form that are not inherited or written in the code of each DNA cell. Drugs are now on the market that either inhibit DNA from being methylated or reroute the mechanism altogether. One such drug by the name of RVX-208 is used to treat patients with cardiovascular disease. This is a prime example of how diseases are not read and produced by ribosomes in DNA, but rather result from lifestyle choices and environmental influences. Epigenetics, though a relatively new field, has profound and important implications on how human lives are affected by induced or unwanted environmental factors. There are a vast number of epigenetic-based diseases not limited to atherosclerosis, mental retardation, and/or cancer. As we move toward a deeper understanding of the molecular mechanisms of epigenetics, we can rest assured that diseases may not only be controlled, but avoided altogether.